Everyone, I think we're going to go ahead and get started. Good morning and welcome. My name is Sarah Hoffman, and I am a scientist in the Applied Biological Services Division here at Biomark. Thank you so much for joining us. We're very excited to tell you about some of the work that we have been doing. We've been very fortunate to remain open. We've established some new safety protocols, but we are still in the office and here to help anybody with anything they may need. So please feel free to reach out to us for whatever your needs are. This morning, I am so lucky to introduce my colleague, Dr. Kevin C., who is going to talk about a lot of the work that he has done estimating abundance and movement of fishes. And with that, take it away, Kevin.
Thank you, Sarah. Let's see.
Oh, I'm going to say one more thing that I totally forgot. Everybody is muted, so if you have questions, please put them into the chat box and we will address your questions as we go. Thanks.
Thanks. Can everybody see the title slide here?
Yes.
Great.
Yes.
So thank you guys for coming this morning. I am a senior biometrician here at Biomark. I've been working on various questions related to salmon populations for nearly a decade here in the northwest of the United States. Today, I'm going to talk to you about a couple different models we've developed to help estimate the abundance and the movement of adult salmon populations. And I just want to acknowledge that my co-author, Mike Ackerman here, has helped a ton with the development of these, and he's also on the call. So great. Let's talk a little bit about there we go. All right. Key question we are looking at today are essentially how many critters are out there. Some of the challenges we're going to confront in answering that include sampling a small portion of the entire population. How do you expand that appropriately? How do you deal with imperfect observations, whether that's observation error or in the case of pit tags, detection efficiency, so you don't detect all of them. The mismatch sometimes between timing of critter movement and when you're collecting data, so that causes you to sometimes miss some individuals. How do you account for that? Sometimes these critters, these fish are moving back and forth, and occasionally you may double count some of them. How do you account for that so that you get an accurate estimate of how many of them are out there? Quick outline for today's talk. We're going to spend the first chunk of it talking about a model to estimate abundance at a dam. This is the State Space Adult Dam Escapement Model, or STADM, that we've developed, and we'll kind of walk through the data that goes into that and how that model works and look at a few of the results that come from that. Beyond that, we'll then look at after the fish cross the dam, when they move to tributaries, and we're trying to estimate how many of them move to different spots, what's the abundance in those tributaries and in those populations. This is often a key question for a lot of fisheries managers. And we have developed the Dam Adult Branch Occupancy Model, or DABOM, to help answer that question. So we'll walk through some of that. And then we'll get into additional population monitoring tools using genetic and age information and how that can enhance your status and trend information and what that looks like. And I should say, if anybody has questions along the way, please feel free to enter them in the chat, and I'm happy to help clarify things as we go. So for today, this is the study area we're going to be primarily focused on. In the little inset map, you can see the Columbia River Basin. That's where all of this work is taking place. That's in the northwest corner of the United States, drains into the Pacific Ocean. The Columbia River Basin drains an area about the size of France, for a little bit of context. And for most of the examples in this talk, we're going to be focused on the Snake River Basin, part of that, which in the larger map is right around this area. And here it is in the larger map. The Columbia comes and the Snake comes in right up here. Lower Granite Dam, the black dot on the map, is the last large dam that these salmon are crossing as they come back from the ocean. I suppose I should mention for anybody who's not familiar with salmon, right, these are fish that spawn in rivers, eggs hatch, they develop as juveniles to some degree or another before headed out to the ocean. They spend, depending on the species, somewhere between 2 and maybe as many as 5 years in the ocean before returning to the river where they spawned to spawn again. So we're primarily concerned, the questions today, with tracking those adults as they return to the river. So these are fish that are swimming in from the ocean. They've traveled the Columbia River, taken a turn onto the Snake River, and by the time they get to Lower Granite, I think that's the 8th dam that they've crossed or something to that effect. So they've done impressive work to that point. And you can see where they may go after they hit that dam into all of these tributaries here. The 2 red dots, Hell's Canyon Dam and Dworshak Dam, are two dams that block fish passage. So fish are not going any further upstream from those. But you can see some of the other tributaries they may move into. The large polygons on the map show Chinook major population groups. So that's one way to organize all of the populations for Chinook salmon. And so answering questions at that spatial scale can be important for managers. And this is where we're going to primarily be focused on for this talk. So let's start with estimating abundance at that dam. So this is focused on Lower Granite. There is, well, yeah, you can see it's a large imposing structure. Fish are trying to get across that on their way upstream. And there are a few different data sources that we're going to use to get an accurate estimate of abundance there. So this is the fish ladder that fish are climbing on their way up and over the dam. I should say that the important questions for management agencies come down to how many wild fish, hatchery fish, and also they have hatchery unclipped fish moving into the Snake River Basin. And so they'd like an accurate estimate of each of those origins by species. So there's 3 primary data sources that we're using to answer this question. The first is the window count. So as fish move up and over the dam, they swim past a window that looks something like this, where human observers are standing there counting the fish as they go by. And they're summarizing those counts by species on an hourly basis. The window is generally open somewhere between 10 and 16 hours a day. And by open, I mean people are manning that window and actually making that count between 10 and 16 hours a day, depending on the time of year. So that's one important source of data. Another one would be pit tag detections as the fish swim up and through the fish ladder. This is just a little diagram showing the top of the fish ladder before they exit into the Snake River. And you can see there's a number of different pit tag antennas located within the ladder to get detections of previously tagged fish. So these would be primarily fish that were tagged as juveniles before they left for the ocean, or possibly coming back as adults at one of the lower dams like Bonneville. And you can get information about, yeah, something about what's going on with those fish when they're crossing the dam and so forth from that sort of information. And finally, there's the adult fish trap. So as fish climb that ladder, the fish trap opens up about 4 times an hour for some specified amount of time, depending on the set trap rate. And if any fish is swimming across the ladder at that moment, they fall into the trap where they can be sampled, they can be measured and pit tagged, and a lot of other data taken within that trap. So those are the 3 main sources of data, and we're going to try to combine all of that and come up with one estimate of abundance. And I'll show you kind of a schematic of how we combine those data and what that might look like. So from your pit tags, you can get a few different pieces of information. You can get an understanding for the proportion of fish that may be crossing the dam while the window is closed, right? So, and by closed, again, that means nobody's actually counting them. So these are fish that are being missed by that window count. What proportion are going over at night? And there is some non-zero portion where that's happening. And that may vary by species and by time of year and so forth. The other thing you can use is between those pit tags and the information from the fish trap, you can get a good estimate of the trap rate for that fish trap. So out of all of the previously pit tagged fish that are crossing the dam, what proportion were caught in the trap? That gives you a good estimate of the trap rate. From that trap rate and the number of fish in the dam, or sorry, fish in the trap, that gives you one way to estimate the total number of fish crossing the dam. Another fairly independent way from that to get at the same number would be looking at the window counts and combining that with the night passage rate. So if they count so many fish at the window, what proportion may they have missed during the nighttime? And that's another way to look at the total fish crossing the dam. So those are two independent sources kind of getting at this unknown but underlying true state of how many fish going across the dam. That's part of the state space part of this model. And from that, you can also estimate the potential observer error at the window. Otherwise, without that, you have no way of putting any uncertainty around those window counts. Now, the total fish crossing the dam is not the total answer, not the end result. There are fish who cross the dam, fall back over it, and climb up that fish ladder again. And so those fish are potentially double counted at the window, so they're inflating the count. They also have the chance to be caught more than once in the fish trap. Again, using pit tags, you can get an idea, an estimate of that reascension rate, and take the total fish crossing the dam and discount that by that reascension rate to get an estimate of the unique number of fish crossing the dam, the unique escapement. And then again, using data from the fish trap, you can look at the origin proportions. So again, those origins would be wild fish, hatchery fish, and hatchery no clip or unclipped hatchery fish. And you can take those proportions, apply it to the unique escapement, and now you have solid estimates of wild hatchery and hatchery no clip escapement, which is what we were hoping for. Another way to look at that is just kind of explains it through some distributions. The window count would be that dashed line in the top left panel here. So how many fish are crossing during the day? As I mentioned, with the fish trap, we can help put some uncertainty around that, which is represented by that red distribution there. You account for night passage, so you're getting a few more fish. Then you discount that for reascending fish, so it gets a little bit smaller. And then you split those total fish into those unique origin proportions, unique origins. And one of the big points I wanted to make with this slide is just the idea that the uncertainty in all of these estimates is captured by sort of the spread of those distributions. And as you utilize more data and account for more of the processes going into this, obviously you're introducing more uncertainty, and you can see that reflected in, by the time you get down to the unique wild fish, that distribution has gotten a bit wider than what we started with. That's a feature, not a bug. I think that's an appropriate estimate of the uncertainty around that. So your confidence intervals are, again, they're appropriate for what the answer is.
Hey, Kevin, we had a question come in.
Yes.
How many years of data do you need in order to continue to improve upon your uncertainty?
That is a great question. We have run this model at Lower Granite for nearly a decade now. However, to date, we generally run it on an annual basis. So it's 1 year's worth of data going in every time. We We run this on a weekly time step. So the nighttime passage, for instance, well, the window count certainly changes week to week. The nighttime passage rate is allowed to change week to week. Reascension rates and those proportions of different origins shift throughout the season. So in that sense, it's somewhere between 30 weeks for spring, summer Chinook and essentially 52 weeks for steelhead, although some of those, there's really no fish going by. So somewhere in the range of maybe 40 weeks with solid data. So if that in terms of the number of data points you're using for any particular year, hopefully that helps answer the question. You could build a hierarchical version that tries to combine different years. We found that 1 year's worth of data seems to provide solid estimates for that year, and so it seemed unnecessary to do that to date. Here's an example from 1 year. This is looking at Chinook, spring, summer Chinook in 2014. The blue circles, which hopefully you can see on some of these points, are the window counts. The red stars are the trap estimates, so number of fish caught in the trap divided by your trap rate with the associated uncertainty as those pink ribbons. And then the Statham model estimates in black with the gray bars showing the uncertainty around there. There are just a couple things I wanted to point out looking at this plot. One is that the window count and the trap estimates generally track each other quite well. However, sometimes the window count's larger than the trap estimate, sometimes vice versa. The model estimate tends to split the difference somehow, somewhere in between, right? So you can see some of that in different places. It's not exactly halfway between the two. Where it falls depends to some degree on the associated uncertainties estimated from the trap or the window. For instance, this week here, this is towards the end of May, beginning of June, the uncertainty around the trap estimate you can see is fairly large or larger, potentially due to not as many pit-tagged fish crossing that week for one reason or another. And so the model tends to track closer to the window counts that week. Also wanted to point out in other cases, like this one in the end of July, there were clearly lots of uncertainty in the trap estimate. You can see those confidence intervals getting very large, and the model is discounting that and essentially tracking the window count very closely that week. The other thing I wanted to point out in this plot was this period towards the end of July, beginning of August, when we had no information from the trap. That year, the fish trap had to be shut down for a period of time due to high temperatures. It was too stressful for the fish. They were already dealing with stress from high temperatures, so it was deemed too stressful to have them fall into the trap to be poked and prodded and measured. So they shut the trap down for a number of weeks, meaning we had no data from the trap. The state space nature of this model handles that missing data, no problem. And you can see the model estimates continue through that time period, and it's using whatever estimates it was able to glean beforehand in terms of the window observer error and so forth to continue the time series of estimates. One of the last things I wanted to point out with this model, I mentioned there's two different processes that are happening that kind of, well, potentially cause some error or bias in the window counts. One is that nighttime passage rate. So there are fish swimming by when nobody's counting them at the window. The other one, so that you're undercounting potentially. The other process is fish reascending the dam, potentially being counted twice. So you might be overestimating fish due to that process. And just looking at how they balance each other out. Right, so this is plots across all the years of data we have. Each dot is a week estimate. And the size corresponds to the window count that week to give you some idea of the run size going on that week. And if the night passage and the reascension rate balanced each other out perfectly, all those dots would fall right along the dashed line. And the point of this plot is just to show that that is not the case. So clearly, sometimes reascension rate is higher than night passage rate and vice versa. And so accounting for both processes independently allows us to get an accurate estimate by week for the unique fish crossing the dam. To wrap up this portion and some of the benefits of Statum, the state space nature of it means it does handle missing data very well. It also incorporates observation error on purpose, and I think that's important. So again, it provides appropriate uncertainty and so appropriate confidence intervals to communicate to managers. It certainly answers the question that we're primarily interested in terms of how many unique wild fish, also how many unique catchery fish are crossing Lower Granite Dam. That's an important question for managers. Yeah, and I think we've shown through simulations that this works quite well. Moving on to the next portion, where do they go from after the dam? So how can we estimate movement beyond Lower Granite Dam into the Snake River ESU? So for the data for this is coming back to that fish trap. As I mentioned, fish are falling in and they're being measured, meaning that all kinds of different ways. You can take the length, you can take the weight, you can take a genetic sample, you can take a scale sample, and analyze all of that after the fact. The wild fish at Lower Granite are also getting pit tagged. And so those pit tags, for those of you who are unfamiliar with them, it's a passive integrated transponder tag. It emits a unique signal when a fish crosses an antenna like these two here in the stream. Right, so it's an example of an array across the river. And when I say array, I'm generally talking about a span of antennas. So one of these lines going across, I would think of as an array. So this is a double array site. And as the fish swims across these antennae, it emits that unique signal, you pick it up, and so you get a detection of that fish at that site. And so those are the detections we're using to estimate movement. This map kind of shows you, again, where all of those detection sites are. So here's Lower Granite again up in the top, and all of these little dots show a unique detection site and its code across the whole Snake Basin. And all of those sites are connected to the system called Patagus that tracks all of the pit tag observations within the Columbia River Basin. So for a given set of tags, you know the tag IDs that went in at Lower Granite. You can query all of their detections and get a sense for their where they were seen across this whole landscape. I think at this point there's over 100 sites going into this model. So we're utilizing data from all of those places across the landscape. Some of them are arrays in the stream. I'd say most of them are, but there are some that are handheld detections, like at a weir, where you're scanning a fish and you get a detection there, and so forth. So we're really trying to utilize as many observations as we can to help with this.
Hey, Kevin, we had another question come in.
Yes.
Is there a minimum amount of detection sites that you need in order to populate this model?
That is a good question. And I would say it depends. It depends on what the size of the area that you're interested in working in. I mean, this one, so the Snake River Basin is fairly large. And after fish cross Lower Granite, they have lots of choices, or they're fish are generally going home. So I don't know if they specifically make that choice, but there are lots of places they could be going. On a much smaller system, and we've developed models like this in smaller watersheds with maybe as few as 10 sites, because it's a much smaller area. You know, maybe it's just a watershed the size of this little circle that I'm trying to draw on the screen right now. So It depends on size of your watershed. It also depends on the number of tags you're putting out, and where they might be going. If you, I think in developing these models, sometimes people have put out sites specifically to help with feed data for these models. And when you do that and you strategically place your sites, say at the mouths of your major tributaries where fish are going, so you get good coverage no matter where your fish may be traveling, you can be strategic in how many sites you put out, for sure. And maybe that'll become clear. I'll show at the end some examples of other places we've developed these models, and you'll get a sense for how many sites are in each of those as well. This is, I would say, out of all the examples we've done, this is probably the largest area and the largest number of sites. So we estimate those movement probabilities using what we've called the dam adult branch occupancy model, or DABOM. And that's essentially a hierarchical patch occupancy model. So thinking about different areas on the landscape as patches that a fish might occupy. And if it's in one patch, it has the chance to move to some of the other ones further upstream. So in this little toy example, here's a fish swimming upstream. So it's going up the river past site A, and it might turn into tributary B, or might go up C, or might go towards D, might stay in that main stem area. There's a detection infrastructure out on the water. So A, B, C, and E are double arrays. So as I mentioned, that looks something like this, where you have two spans of antenna. So essentially two chances to detect the fish if it goes past that site. Site D, for an example, might be a weir, where you're scanning the fish by hand, or it might be a single array, so you essentially have one chance to spot that fish. And so, yeah, it incorporates, so this model is going to incorporate that branching structure of the stream. As the fish moves past A, where does it go? And so we're going to estimate those movement probabilities. Moves past A, there's a multinomial distribution of, it has to either go to B, to C, to D, or to this gray box, this black box here, which is essentially fish that have crossed A, but haven't gone past any of those other sites. So maybe it spawned in the main stem area there, maybe it died there, maybe it was harvested, caught by an angler. And then the other movement probability in this example would be moving, if you go past site C, you make it all the way up past site E. So those are what we're really aiming to estimate. That's the key here. And getting at some of the, say, the probability of going from A all the way to E is just a matter of multiplying some of those probabilities together appropriately. So you can imagine you incorporate a lot of these movement probabilities across that vast network of sites, lots of different multinomials, or in this case from C to E, a little binomial, did it make it past E or not? And then you multiply those estimates appropriately and end up with a chance that a fish moves to any particular site. You take those probabilities and multiply them by your total escapement that you started with. So this goes back to how many unique wild fish crossed Lower Granite Dam. And we got that answer from Statham. And so I should have mentioned in this example, all of our tagged fish are wild fish. So when you multiply it by that total abundance, you turn these movement probabilities into estimates of abundance or escapement past each detection site. Now, in the process of doing this, the model is also estimating detection probabilities. So what are the chances that if the fish moves past site B, that you actually see it? And what are the chances you see it on each array? Right? So there's two detection probabilities at site B because it's a double array. And so you can estimate that for A, B, C, and E, because those are all double arrays, or at least have an upstream detection somewhere. At site D, where you have one chance to see the fish and there's no upstream detections, there's not enough information in the model to estimate that detection probability. So we're going to have to fix it. Often you can fix it at 100%, which leads to a conservative estimate of escapement past that site. For these adult salmon, that's often not a terrible assumption either. Most of the detection probabilities that we estimate are in the 90% or higher range. And the further upstream you move, so the shallower the river, the higher the detection probability generally is. And often these weirs or those single arrays can often be high up in the tributaries. The other thing I should mention is that if it's a weir, and if you have some independent estimate of the capture probability there, so perhaps the people running the weir are running a mark recapture experiment there, and they estimate that that weir is operating and catching 85% of the fish going past there, you can plug that estimate of 85% into the model as a prior. This is running a Bayesian framework. So you can utilize that instead of fixing it at 100%, put in the estimate of 85%, and that will improve your accuracy. Because one of the things that's going on within DABOM is it's estimating those movement probabilities in the context of those detection probabilities. So for instance, if B happens to be a terrible site and is only observing half the fish moving past that area, if you can estimate that the detection probability is actually 50%, and you can do that well, the movement probability going into B will increase compared to if you assumed it was 100% detection, right? Because the model is going to recognize that there will be fish slipping past B undetected, and so it'll increase the probability of fish going there. Hopefully that made sense, but I think that's one of the key underlying features of this model is that those movements are related to the detection probabilities, and the two are certainly intertwined.
Hey, Kevin, another question came in here.
Yes.
Does the model allow for backward movement, i.e. if the fish enters multiple tributaries?
Great question. I was about to talk about that. So give me a sec, give me about 30 seconds and I will get to that. Because this is an example, this is what the data looks like. It goes into the model. It's capture histories, it's ones and zeros. In this case, for our little toy example, each of those double arrays has a column. So the two arrays in site A correspond to A1 and A2, and you might see the fish or not on each array, and so forth and so on. And so that's the data you're plugging in. I just wanted to point out that in this case, here's a fish that went past A, but was not detected there at all, but was seen somewhere up at site C and even at site E. So you know it must have moved past A undetected. That information helps the model estimate the detection probability at A. So it's not just the two arrays there, but it's also all of the upstream detections that help get you the estimate of detection probability at sites. Now, there are cases where you have a fish, in this case, came past A, poked its nose up into tributary B, whoops, and then came back and went up tributary C. The model can't handle that sort of movement. It does assume that fish are on a one-way trip, moving up, taking only one path through that stream network. So we do have a program attached to this that helps identify those types of capture histories, and it takes somebody looking a little more carefully at the observations to make a call. So somebody would go in and look at this fish and say, okay, when was it seen at site B? When was it seen at site C? And obviously these capture histories are kind of a summary of was it seen at site, at site B at all or not. So there's potentially lots of detections on each array in reality that gets summarized into just a 1 or a 0. So you may be able to tell that, oh, this fish moved up. You might be able to see the movement, moved up B and it moved back down 2 days later, and then it went up C. Or it moved up tributary B, it stayed there for 3 months, and then it came down and went up C. And so based on the timing, based on knowledge of that species' life history, a fisheries biologist would need to make a call about which tributary do we think it spawned in. And based on that, you would delete the detections from that other stream before feeding it into the model. So hopefully that answers that. But yeah, the model does have to assume that all these fish are on a one-way trip. Obviously, that's not always the case. You see some fish bouncing around, particularly with steelhead. So there is some data cleaning that has to happen before running this model. The output, as I mentioned, you get estimates of abundance or escapement past every single detection point, right? It's just a matter of doing those movement multiplications correctly. So you might be interested in this entire area in the upper Grand Ronde, or you might be focused on a particular tributary there and the escapement to that particular spot. And you get all of those as output from this model. You can imagine those circles spreading across the whole map here. And you can add up different things together. So you could add estimates for all the populations within one of these major population groups, for instance, if that's your question of interest. And again, you can do this in the Bayesian context. You could do that and get appropriate levels of uncertainty, given that some of those movement probabilities are correlated. That can all be handled. So that's great. One of the other benefits, or one of the other features we've built into this, particularly for this version in the Snake Basin, is the idea of having time-varying movement parameters, particularly for fish just after they leave the Lower Granite Dam. The reason behind doing this is, and so the idea there would be that the movement probabilities for those initial branches might change through time. So it's not fixed for the entire season. You can see these are the results, the estimates for some of these. The idea that, well, if you're earlier in the season, you have a higher chance of going to the Grand Ronde, this green line here, versus later in the season, fish might have a much greater chance of going to the South Fork salmon. Why that's particularly important here is that the trap rate changes at Lower Granite Dam. So the sample of fish that you collect over the course of a season is not necessarily representative of the run at large. That trap rate changes for a variety of reasons. Sometimes it's just shut down because of temperature concerns. Sometimes it changes for logistical reasons as you get different species moving through with different run sizes. Sometimes you have to turn the trap rate down so that the trap's not flooded with fish. There's only so many that the people working there can handle. Other times, if there's fewer fish, you might have an interest in increasing the trap rate. So there are times where it changes. And the idea here is that interacts with run timing from different populations. So if the trap rate goes from a higher level to a lower level later in the season, the fish moving to populations, some of the fish coming back are more some of the populations have differential run timing, right? Fish coming back to Grand Ronde tend to show up earlier. Fish coming to the South Fork salmon tend to show up later in the run. And so your South Fork salmon fish will be underrepresented if the trap rate had dropped later in the season. So allowing those movement parameters to vary through time helps correct for that. If your trap rate remains constant through the whole season, then you don't have to worry about this, and it does make it a little bit simpler.
Hey, Kevin, we have another question about the DABOM model. Is it reliant on pit tag data, or could other data sources such as acoustic or radio telemetry be used to populate a similar model?
I don't see why you couldn't use acoustic or radio telemetry tags or any other detection technology to run a model like this. It's reliant on those capture histories. As long as your your marked fish, whatever fish you tag with whatever method, are representative of the population at large. So as long as you meet that assumption, I think you could certainly do this with other technology.
I think that addresses another question that came in as well, which is, can you do ichthyological monitoring without necessary capture of fish? So to your point, could something like an observation tower be used to populate a model?
I would say not this kind of model, because this one relies on individual detections or detections from individual fish. So it's those individual capture histories of this fish moved to this site and then to this site and then to this site. And if all you had were counts from something like an observation tower, that would not provide you the information for it. There may be similar but different. I mean, there may be ways to tweak this, and tweak makes it sound like a small thing, but I think you might be able to take the general framework in terms of that branching network and make that correspond to counts. That would be a really interesting question and a really interesting problem. You might be able to do that. I would be fun to work on.
Another question has come in for you. How do you account for unmarked hatchery fish?
How do you differentiate those unmarked hatchery fish? So you can't tell by looking at them. That's done through genetics. So when you take a genetic sample of all the fish in the trap at Lower Granite, they have a genetic record of all of the hatchery parents in the basin, and they match that up, and you can tell, oh, this fish came from this hatchery. This fish is not in the record, so it's a wild fish.
Excellent, thanks. I think that's all I see at the current moment. Thanks, Kevin.
All right, no problem. Keep them coming. Let's talk a little bit about some validation work we've done, just trying to prove that this thing works. So this was information we compiled from a variety of weirs all around the Snake Basin, considering estimates at weirs, they're completely independent. We also figured they were pretty rock solid and kind of a good truth to compare our estimates to. So the weir estimates are in blue on all of these time series plots. The pit tag based Statham and DABOM estimates are in red. And in general, I just wanted to point out the two track each other really well. You see the same trends through time, the same shapes in terms of ups and downs. Most of the weirs, you see the points practically overlaying on top of each other. There are a handful that don't match up. And in doing this exercise, which we did this a couple of years ago, it actually helped identify there were a couple weirs where people had scanned all the fish and they'd kept all of those records on their local computer, but had forgotten to upload it to the overall database, Pitagus. And so obviously the DABOM model relies on those observations, and your estimates are only going to be as good as the observations you feed it. So when we identified that giant mismatch in some of these places, people found those records on the computers and uploaded them, and things started to match up much better. You can also put this on just an XY scatter plot and see most of those points clustering around the 1-to-1 line. And for both species, I'd say it really does match up quite well. In particular, these weirs are often pretty high in the watershed, so fish have traveled a long way from Lower Granite by the time they get there. And we're looking at not that many pit tag observations for any particular weir. And so the fact that it still provides pretty comparable, very solid estimates is great. So to wrap things up around DABOM, so one of the important things I think about it, as I mentioned, you do have some weirs scattered around the Snake Basin. However, they're not cheap to operate, and you can only put them in places where the river's small enough to build a weir. and so forth. You can put in a pit tag antenna in many more places, and it may be cheaper to operate. So it may be cost effective and help provide much greater coverage. Particular in the Snake Basin, it can be very hard, particularly for steelhead, to get an estimate of adults because they're coming into those tributaries during the springtime when there's spring runoff, makes the rivers really cloudy, they're running high. It's dangerous to be out on them in many cases. And so these kind of pit tag detections may be your best bet to actually estimate how many fish are moving into those tributaries. I think we've shown through that validation work that it works, it's accurate. It does incorporate all the sources of uncertainty. So that detection probability, the uncertainty certainly changes depending on how many tags you observe. So places that see a lot of fish, you'll be much more certain about. Places that only see a handful, you'll have wider confidence intervals. But I feel like, again, that's a feature, not a bug. That's an honest assessment of your uncertainty. And when you do the multiplication through of movement times total escapement at Lower Granite, it does incorporate the uncertainty in your Statham estimates from Lower Granite. So all of that's coming through to your final abundance estimates. We talked a little about those time varying probabilities and how that can be useful, and how you it helps you get estimates on a variety of spatial scales depending on your question of interest. Some managers might be interested in as I mentioned, the major population scale and the status there. Other people might be interested in a particular tributary. You get all of that. In other places, you can use both hatchery and wild fish at the same time. Plug those into a model like this. You can use both of them to help estimate the detection probability. Obviously, that shouldn't change depending on hatchery or wild fish. However, hatchery fish are clearly trying to go back to their hatcheries, which may not be in the same places that wild fish are going. So you can separate their movement probabilities, and you end up with very different abundance estimates depending on where they're going. Let's see, I guess I should have pointed out towards the beginning, people were talking about how many, some general ideas. So for Lower Granite, we tag somewhere in the, I think it's about 2,000 to 4,000 fish, depending on the year and the species. Generally end up tagging more steelhead because they potentially go to more of those tributaries. There are some places that they just won't, Chinook don't go, and therefore we don't have to worry about those detection sites. We've also determined, if you're thinking about setting these up, that you need, if you wanted an estimate of escapement with a coefficient of variation less than 15%, so pretty confident in your estimates. That confidence level seems to correspond to the number of detections you get at that site. So you'd be hoping for somewhere in the range of 40 detections at that site, 40 fish, which might, if it's a site with high detection probability, you just need 40 fish to get past there. If it's a site with low detection probability, you obviously need more fish, more tags to move past there, but in the end, you're hoping to get 40 hits. Hopefully that helps and gives you some context. Again, I think that number can change depending on the size of your overall watershed, your overall basin, where how many places the fish are trying to go, and so forth. So let's talk about some of the different applications we've built for this kind of model. We've been talking about this Snake Basin model, starting at Lower Granite. We've done that for wild spring summer Chinook and wild hatchery for a number of years. Wild steelhead. The Upper Columbia for fish starting at Priest Rapids. We've done this for wild and hatchery steelhead for a number of years. We're going to start running this model for wild and hatchery spring Chinook, and also wild and hatchery coho for that area. We have built a smaller model for spring Chinook in the Upper Wenatchee, so that's kind of a sub basin up here. We've done that for a number of years with fish that are tagged at Tumwater Dam. And recently, we've also built a model to estimate wild steelhead in the Yakima Basin, fish starting at Prosser Dam. So, variety of places, they all have that sort of branching stream network. All the black dots are those detection sites. So you can see you set those up across the landscape. You obviously need to build a different branching structure for each spot, but it can be done, and it seems to be working quite well. So that was all about getting abundance into these tributaries. I wanted to spend the last bit of this talk talking about other data that you can bring to bear to help your population monitoring and create a fuller picture of the status of your populations. So this goes back to those fish that are tagged in the fish trap, right? They're tagged with a pit tag, and we've been talking about how you use that data. But I mentioned there's lots of other data that you can take when you have that fish in hand. Lengths, scales, genetic data, and so forth. And you can keep all of that or compile all of that data. You know what all of those measurements are and how they're associated to a particular tag. Then after the fact, you see where that tag went. And so you can compile, here are all the tags that went to the Lemhi River. And let's go back and let's look at the length information there, build a length frequency for the Lemhi. Or here's an example looking at sex ratios, right? So using that genetic information, you can determine male or female pretty reliably. Here's just kind of shows the separation for some of that genetic data. I should mention it, for at least for lower granite, you probably really need to use the genetic data because the fish aren't, they haven't expressed themselves physically in terms of males or females yet in their spawning journey. So it's really hard to tell by eye. With the genetic data, you get that. As I mentioned, then you know, oh, great, out of the 100 fish that went back to this population, 60 of them were females. That gives you a sex ratio, right? And so this is just an example. This is estimates of female proportions by population. They're kind of colored by major population group across all the different years. And so you can take those female proportions and multiply it by total abundance. And now you're looking at total female abundance, which from a conservation standpoint, that might be your particular metric of interest. That could be really useful. The other thing you get from scale data is you can get an age proportion. So this is looking at steelhead. They're coming back at a variety of ages. You can see down at the bottom, some of them come back at age 3, 4, 5, 6, 7, or even 8 years old. And for each population, you can get an estimate of what that age breakdown is for that returning year. What's nice about that is you can use that to tie it back to the brood year for those particular fish and looking at population productivity and lifetime reproductive success. So the plot on the left here just shows a time series of something like recruits per spawner, right? Which is a measure of your population's productivity. For the population to be growing, you need those recruits per spawner to be greater than 1. You can see for this example, most of the time it is. How do you look at that? Well, so this table on the right is data from that DABOM model that we've just been talking about, compiled for steelhead in Big Creek, the Big Creek population in the Middle Fork salmon. We've been talking about how you get estimates of spawners. So that's the total returns for that particular year. But you also then get estimates of the age breakdown. You tie that back to its brood year. So fish returning last year, you break out, great, we had 923 fish coming back. We have proportions of ages, so you can compile or turn that into how many returning offspring by age. All the fish that were 5 years old, well, they came from a brood year back here in 2005, right? And so kind of work backwards like that. And then you can compile after a number of years. Here's all the recruits that managed to make it back to Big Creek from the spawn year of 2011. And so you can look at how many recruits per how many spawners were there that year. and you can generate a recruits per spawner number. This is really crucial in looking at like population viability analysis and so forth. It's a great metric for assessing a population's health. It does take a number of years to completely fill out this table, right? For instance, some of these later years, we don't have the returns yet for age 5, 6, 7 from brood year 2015, right? So this 0.02 recruits per spawner that's not applicable yet. Some of these other ones, the higher ones, yes, this really is 0.75, which is not great, right? We'd like to see that up above 1 for these threatened or endangered populations to be bouncing back. Some of the other added information you might get, this is in particular looking at genetic data from those fish in hand at Lower Granite. You can look at your population structure genetically. You can see which populations are most closely related to which ones are very genetically distinct. You can kind of see that information in this diagram down here. I also just wanted to talk about the mixed stock analysis, the idea that from that genetic data, you get some idea of where these fish may be headed to. And in the DABOM model, we talked a little bit about those black boxes of fish that move past the site and then are never seen again. There is a large proportion in the Snake Basin of fish that move past Lower Granite and are never detected again, potentially somewhere in the 40 to 50% range. Sometimes that's because there are major tributaries, major rivers in that basin that aren't wired up with pit tag antennas. So if fish go there, we won't detect them. In other cases, it may be that fish are caught in hatcheries, or sorry, in harvest. At least for hatchery, maybe wild fish are caught and released, but they suffer some latent mortality because of that, and they just don't make it back to their tributaries. The genetic information at Lower Granite might help tell us about where those fish that are never detected using pit tags, where they might have been headed. And that could help in a run reconstruction type framework, provide some additional information for managers. Wrapping things up, some general take-home messages. I would say all the tools we've talked about this morning are great ways for monitoring status and trend. Talked about how you can use them to estimate abundance in different populations, a sex ratio and age data, and how you can turn all of that into lifetime reproductive success and recruits per spawner, which are great metrics for population viability analyses. I've mentioned a number of different times how we're trying to incorporate all the different sources of uncertainty to provide appropriate confidence intervals. I think that's important when communicating to managers and help them understand what not just the point estimate, but yeah, how confident are you in that estimate? A lot of this information has been well validated, showed some of that, so we know it works. As mentioned, we've used it in multiple basins for multiple species, and that's been great. And if anybody is curious about what it might look like to implement something like this in your basin, I'd love to talk further about it. My contact information's here. There's my email. Please feel free to get in touch. And I'd also be happy to take some more questions at this point. Thank you.
Thanks, Kevin. Another question that's come in, could you maybe talk a little bit more about the use of aging in relation to monitoring abundance in salmon populations?
Sure. Anything in particular? I think the key point about aging and age data is that it allows you to tie those returning adults back to a particular brood year. So you know that the fish coming back this year that are age 4 came from brood year 4 years ago. And the ones that come back this year that are aged 5 came from the previous brood year, right? And so it goes back to building that sort of spawner recruit table where you can start looking at, okay, how out of all of the spawners from 2010, some of them came back 3 years later, some of them came back 4 years later, 5, 6. In total, we got, over a number of years, we got 696 returners from that brood year. Obviously, for a population to be growing, you'd like that to be, you'd like this number to be larger than this number. But all of these populations are, that we've been talking about, are endangered or threatened. So there's a reason that maybe that recruits responder is less than 1. But I think that's the key point to why aging is important, particularly for steelhead. As you can see from this, they come back at a variety of ages. They're they might leave for the ocean at a variety of ages and spend at a variety of time out there, and so tying their returns back to a particular brood year can be really important. And the only way to do that is with this kind of age data. I guess I should also mention the fact that you get those fish in hand, in this case at Lower Granite, and you can take that data that way. I think that helps with the assumption that, obviously you're not aging every fish, not aging all 923. You're aging however many fish that were tagged and were detected going to, in this case, Big Creek. So you need to assume that that's a representative sample of all the fish that went to Big Creek. I think the operations at Lower Granite allow you to meet that assumption potentially much better than relying on, say, carcass surveys within Big Creek or something so forth. I mean, I've done a little work on how carcass surveys you may be biased by sex or size or other ways. And so I think this method shows some improvement on that, but you also potentially get that information for all of your populations above Lower Granite.
Excellent, thank you. There are a couple other questions that came in that are a little more relative to salmon biology in general. So the first is, does nighttime escapement have any consequences in relation to photoperiodism?
Somebody would need to tell me what photoperiodism refers to. I'm sorry.
I believe that it's a behavioral response to the change in day length. So is there a seasonal effect on nighttime escapement?
As in, do you see a difference in that nighttime escapement rate through the year that might correspond to changes in daylight?
That seems like a good interpretation.
That's a great question, and one I haven't looked at specifically. I will say that the change in, at least at Lower Granite, the amount of time that people are counting at the window, as I mentioned, it changes from 16 to 10 hours. The 10 hours happens to correspond with generally the late fall, winter, and early spring period, in part because there are fewer fish going past Lower Granite then. So they're cutting down on man hours. That obviously would also, of course. So let me back up. That means that the night passage rate, I mean, it really now, instead of looking at, okay, how many fish are crossing during the 8 hours in the summer when the window's closed, changes to how many fish are crossing during the 14 hours in the winter when the window's closed. And so the rate, I would expect it to maybe get bigger in the winter anyways because of that reason. It's just the meaning of that parameter changes slightly when the hours for the window operation changes. But I haven't really looked at whether that has a biological cue as well. But you certainly could.
Thanks. And then one more question that's, again, perhaps more related to salmon biology in general. Are there habitat restoration actions that are used to improve living conditions, particularly in relation to dissolved oxygen, such as the development or implementation of blocks and piles? Can there be a negative effect where it creates dead zones behind those blocks or piles, I assume, that negatively impact the survival of salmon?
That's a very legitimate question. It's not one I'm as familiar with. I'm imagining that's maybe we're talking about blocks or piles in the main stem area, so maybe through the migration corridor for some of these. And honestly, that's probably out of my area of expertise that I'd want to say anything about.
All right, well, that's all that I see coming in right now. If anybody else has any other questions, feel free to put them in the chat and we'll stay on for another couple of minutes. Otherwise, thank you so much, Kevin, for speaking with us about your work. And we really appreciate everyone on the call today. Please feel free to reach out to us if you have any additional questions. We'd be happy to speak with you more about this type of work and others that we are doing within Biomark.
All right, thanks guys. I think we're probably good. Sarah, did you stop the recording?